Hello, everyone, and welcome to week 3 of the Medical Research Scotland Academy. This week, we are delighted to be joined by Professor Heather Whalley, who is Professor of Neuroscience and Mental Health, as well as being Chief Scientist for Generation Scotland at the University of Edinburgh. We're also going to be live from the lab with Lee Murphy, who is Head of the Genetics Core at the Edinburgh Clinical Research Facility at the University of Edinburgh. And we'll be meeting his team of research technicians, including Emma Aitken and Richard Clark. We'll be sharing some really interesting background on how data is gathered and used in medical research. And if you're joining us live today, you have the opportunity to share your questions for our speakers using the Q&A button, which is at the bottom of your screen. We have some time at the end of the session for us to answer your questions. So, I think let's get started, and I will hand over to Professor Heather Whalley.
Thanks very much, Sean. So yeah, it's very exciting to be part of this webinar. So my name's Heather Whalley. I've got my email address there, and if anybody wants to kind of reach out and ask questions offline, you're very welcome to do so. So I'm going to talk about the power of big data in healthcare research. So just before I start, I thought it would be perhaps a bit useful to talk about who I am and where I got to where I am at the moment. So I did my undergraduate degree in neuroscience, and then I did an MSc and a PhD in brain imaging research. I then got interested in these large data sets and how they can help with healthcare research. And I'm now Professor of Neuroscience and Mental Health at the University of Edinburgh, and I'm Chief Scientist for Generation Scotland, which is a research platform I'm going to talk about a bit later on. The concept of big data was coined in the 1990s, and it's been developed in parallel to the computing power that's been able to process and analyse it. And as you can imagine, that even since 1990, the concept of big data has really just increased exponentially. So just as a bit of fun, as an example of really why we need computers to analyse this kind of data. So would you like to guess how many 4-drawer cabinets, filing cabinets, that would be needed to store worldwide health data? So 1 million, 2 million? And I think you need to go even higher. And we're talking about 500 billion 4-drawer filing cabinets to be able to store health data. And that amounts to about 25,000 petabytes of data. And that's really why we need computers, because it's just impossible to look at that size of data manually. So moving a little bit more towards healthcare data particularly. So healthcare data includes things like patient data from seeing your GP or going into the hospital. It includes things like blood or tissue samples that we extract information from, lifestyle data, brain imaging data, and genetic data. And it includes things like data from health and backgrounds and lifestyle, and from health and fitness devices. And big data in healthcare research refers to the gathering of this kind of information and linking it together so we can make new discoveries about people and their health. So using the power of big data analytics to improve healthcare for everyone.
Good.
So this is just an example of what healthcare data might look like. And this slide shows an example of prescription data, and it's just a snapshot of a small number of patients and a small number of drugs that are taking the medication, but it's actually based on real world data. And this is kind of the example of what we can see when we look in the healthcare record. So you can see on the left-hand side, we've got patients 1, 2, 3, 4, 5, and 6, and the different drugs in the different colours along the bottom axis, and the time periods that the individual patients are taking these medications. So you can see from these that patients 1 and 2, for example, are taking just one medication and then for a brief period of time, and then they come off. But we can see patients 3 to 6 are doing lots of switching between different medications for the same condition. So what can we infer from these kinds of patterns? Well, we could infer that since patients 1 and 2 are taking the medication and then coming off, that they might be categorized as responders. But patients 3 and 6, they keep switching. They might not respond to the medication. That's maybe why they're switching, or they might have complications with taking the medication. And some of them might not even find medication that works. And we refer to these as non-responders. But to put that into a clinical context, so for example, if you're a doctor and you're seeing a patient, you would really want to know which drug is going to suit which patient without them having to go through a long switching process, which is not just costly, but it's time consuming for the patient as well. So what we really want to be able to do is find a way to match the drugs that work to the people that will respond to them, to avoid them having to go through these periods of switching medication. So one of the things that we're doing in Generation Scotland is that we're trying to link prescription data to genetics. So we can try and use genetics to predict who are the responders, and then be able to target treatment to those responders. And it will also allow us to think a little bit more about the underlying biology between those who are responding and those who are not responding. So it's really showing the power of linking genetic data to prescription data to try and make sure that treatments are targeted for individuals who will best respond to them. So, Generation Scotland, I'd just like to give you a brief overview of what this research platform is. So it's a cohort study that was designed in 2006, and it was to improve the health and well-being of people across Scotland. As I mentioned, it started in 2006, and we now have 30,000 volunteers across the whole of Scotland. And we have the data from the volunteers has been used by researchers worldwide, and we've contributed to over 300 scientific papers. And the research areas that we have looked at include mental health, COVID-19, chronic pain, heart disease, diabetes, asthma, and others that are mentioned there. In 2022, we were actually given more funding by the Wellcome Trust to begin to include younger people in research. And that's what I'd like to go on to talk about next. So why do we need young people to join us in our research? Well, first and foremost, young people are underrepresented in research. So if you can look at the graph that's shown on the slide there, so the green line actually represents the peak onset for mental health conditions. And you can see it's covered in the red box, and it's around 15 years old. But the most large-scale health studies in the UK and across the world look at diseases of older age. They tend to look at things like dementia and stroke, and they tend to recruit people who are older, in the older category, so 40 plus. So people included within the blue box. So it's really missing this crucial peak of health that's happening in adolescence. So in Generation Scotland, we want to make sure that we're capturing this early peak for onset of different health conditions, including mental health. And so that's why we're recruiting individuals who are age 12 and above. And we want to learn more about things that affect young people and follow them over time to understand risk factors and ways that we can treat and prevent disease. The second reason why we want more young people in research is that we want to make sure that we're asking the right questions. So I'm just going to give an example of a research study that we did recently, which was looking at teenage irritability. Not just because I think sometimes teenagers get a bad reputation for having that kind of sense of frustration and angst and irritability, but also because it can be a sign of problems with emotional stability. So we were interested in irritability, and we were interested in looking in the literature to see how that had been studied previously. And what we found was that in the literature, most researchers in the past had actually used a paradigm, so a test where they were giving young people a complex mathematical equation that was just not solvable in order to kind of initiate that sense of irritability. But we really didn't think that would, first of all, make all young people irritable, And more importantly, that it didn't capture the real world kind of social context of what irritability feels like. So we went back to young people to ask them about things that make them irritable in their everyday lives. So being nagged to tidy a room, or people queue jumping, or Wi-Fi dropping. So we got them to kind of write some of those scenarios that they found were irritable or initiating an irritable feeling. And then we used those scenarios in the brain scanner to see which areas were activated once individuals were feeling those senses of irritability. And I really just use that as an example to show that it's really important to be asking young people the right questions for health research. So if we'd have gone with the mathematical equations, that wouldn't have included any social context within the study. I've also put a QR code there at the top left, and it's also on the following slide. And this just, if you scan the code, this is how you can get involved in Generation Scotland. There's also some other contact details there. So you can go to www.gen.scot, or you can scan the QR code if you're interested in joining. I think the team is also going to send some information about other opportunities that we have within Generation Scotland to join us, to join some youth advisory groups. So we've got many opportunities. And the next part of the session will be handing over to Lee's lab, where we are going to see the extraction of DNA from saliva samples. If you remember, I linked up genetic data with prescription data to show the power of how combining those different types of information can help. And within Generation Scotland, we use saliva samples to get the samples of DNA. And these are some of the opportunities to join Generation Scotland. And I think these are going to be circulated a bit later. Thank you.
Thank you so much, Heather. That was really insightful. I think you did such a brilliant review of the power of big data and how it's directly applied within the industry. As you say, we're now heading over to the lab to hear from Lee, Emma and Richard about their use of those samples and to find out about the career paths that led them right here today.
Welcome to the Genetics Corps. We are a University of Edinburgh laboratory and joined with NHS Lothian. And we're here to do the kind of genomic studies for lots of different projects within Edinburgh. So we don't do our own research. What we're here to do is provide a platform to allow researchers, either doctors in the hospital or researchers within the university, to get access to cutting-edge equipment like the sensors you can see behind me. But most importantly, to get access to really well-qualified technicians like Emma and Dickie today, we're going to speak to. So Emma, do you want to introduce yourself and tell us a little bit about how you became a research technician?
Hi. Like I said, my name is Emma. I am one of the research technicians here in the Genetic Core facility. So pretty much how I got here. Well, I did my degree in biomedical science anatomy. It was an uncredited course, which is mainly more focused towards research. So got my degree and got my first job out of uni, which was actually a job that was mixed between clinical work and research work. So I did that job for a wee while. So I got my taste of both clinical and research. And this is when I was going what am I more interested in? What is it that I like? Turned out I liked a bit of both. So as the years went on, picked up some transferable skills, got my experience. I kind of did shift over to a bit more of the clinical side of things just to see. And then as the years went on, I kind of went, actually, I think I want to go back to research. I do quite enjoy, I see, the clinical side of things and how this research contributes towards, the clinical side and all the progress that it made. So in a nutshell, that kind of basically what ended me back here to kind of get a bit more involved into the sort of research aspect of clinical studies. And like I say, the good thing about this job is, like you say, we do it for a lot of clinical research projects. So I get to be involved with not just one project. I get to be involved with a lot of things.
So if we focus on Generation Scotland, and what is it you do for Generation Scotland?
So specifically for Genetics in Scotland, my role is basically more in terms of the extraction side. So I've got some examples of samples here. So this is how we receive them. It's the saliva tubes. As we say, we get saliva tubes sent to us. So my job initially will just be, receive them, unpack them, book them into what's called our LIMS system. So this is just basically a way for us to see how many samples we've got, what samples, and track them as they go through each process of our lab. So
I can see on the samples, there's no names on there. It's all anonymized.
Yes. So we anonymize every single one of our samples. So we do this because basically, it's to protect the privacy of our participants as well. I know a lot of worries, like from me speaking to people, they were a bit like, we're giving you quite close data of ours. We're basically giving you all our genetic information. What are you going to do with this? Well, other than research, nothing. It's like we don't know anything about you. We don't know whose it is, what it is, where you're from, nothing like that.
So there's a lot of regulations around working with DNA that we have to abide by. So one of those is that if we don't need information for our work, then we shouldn't have that information. So we don't get any personal information with the samples. They're just barcoded and anonymized.
So this basically just saliva in a tube, really.
And then what do you do with the saliva to extract the DNA?
So to get DNA essentially out of this liquid that you see here, we go through a process of DNA extraction. So basically what we need to do first, we've got our white blood cells that are in here. But in order to get to our DNA, we need to break these cells open because we've got our cells and our DNA are in the cells. So we need to break this open through a process of cell lysis. So that's usually with a detergent of sorts, many kinds, and this will help break the cell membrane down, and this will release the DNA into solution. And through the process that we use through our magic machine, that we call it, we take advantage of the fact that DNA is negatively charged, and we have what's called our magnetic beads. So basically, they attract the DNA out of the solution, give it a few washes, usually with ethanol, just to get rid of all the other impurities that we don't want in the samples. It's just like proteins, like all the contaminants, salts, other things that we don't want to bother with. Give it a few washes, and then we elute it into...
And you got that.
Yes, and we elute it. So we can do up to 96 at a time. So this is an example here of a full plate of 96 samples.
You can see that. So each one of these wells is one person. So you've got 96 people on a plate like that. So you can see from the tubes we showed before, we really kind of miniaturized down the saliva into smaller amounts of DNA. Then we can store those in our freezers.
Yeah, so we can store these for, up to a few months or years. And then this will get processed onto the next stage, which will be Dickie's part.
So we... within the lab, we mostly collect blood from patients, but Genetics Scotland collects saliva. Why would they collect saliva out of blood?
So saliva, although you may necessarily get, less yield from blood, I know kind of like blood is, you get more and make it the preferred way to go. But the benefit of saliva is, people can do it in the comfort of your own homes. You don't have to make an appointment and come in somewhere to get it done. They can do it in their own time. It's easy enough, and plus it's very much less invasive. I know not everyone likes getting blood taken, so, and plus like we're touching on there, we are hoping to recruit a lot of younger people, and I think it would be a lot less scary for them to say, get saliva in a tube rather than a needle.
Okay, thank you, Emma. So I'm now going to ask Dickie to come in and sort that scenes. Dickie, we've heard from Heather about how she started out as a research technician. Your tree was a little bit less linear. Do you want to say a few words about how you became?
I never intended to be a biologist or a technician. I did enjoy biology at school, but it wasn't my first love. I left school and I had numerous jobs. I worked as a chef, nightclub for a while. I became really interested in the world and how it works. So I went to university to do ecological and environmental science. Enjoyed the molecular biology part of it. When I left, I needed a job, and Lee's Lab was offering a I've got temporary position working on Generation Scotland. It's a 3D. Or didn't even think that far back, and then when a fixed position became available, I realised how much I enjoyed working within research, and so the rest is history, right?
From nightclubs to all that.
Absolutely, yes. With all lots in between, I have got this.
Okay, and then what do you do in the team?
Okay, so whereas Emma isolates DNA from primary samples like blood or from saliva, we run a genetics analysis service here as well. So we have arrays for genotyping, and we have sequencers, which are very flexible machines. We can get a lot of information.
So yeah, these are the DNA sequencers behind us. These are called Illumina NextSeq 2000s. So we can sequence DNA and RNA on these. They're kind of medium throughput kind of platforms. So they can sequence about 3 human genomes in a day. The platform, and we've got 2 of those. We don't do many whole genomes in here. Usually we're kind of focusing in certain areas. I just, off the back of that, and we've got some flow cells here that these are what you would ultimately load your DNA samples. Oh, we need one there. That's it. The smallest. That's what we run the DNA on. The DNA goes onto the slide, and then they get fed into the machine.
Great.
And then we have different size cartridges, and I don't know if you can see there, the different sizes and determine the throughput. So the size of the glass slide allows you to add more leads on.
So these sequencers all work. Essentially, you have to take your DNA, which is a very big molecule, and chop it up into small bits. Put special molecules onto it so the sequencer will recognize them, and then it'll read out the sequence of your DNA or RNA. It's a great technology. It's really high throughput. As we said, it can generate a lot of data. But there are also new options. We have a sequencer in a box. So we'll just take it up, This is called a MinION. And it's just a test cell that's in it just now, but
yeah, you camera. So there it is. So that's a handheld sequencing device, which is a little bit different to the platforms behind us. So you can see, this is quite an exciting technology. It uses molecular pores, which are basically protein rings. The DNA goes through the ring, and the protein detects the shape of each individual base, the A's, C's, T's, and G's. And by changing the shape of the nanopore, it changes the voltage signal. That's what's detected from this. And so you can imagine these are very mobile. You can imagine ways they could be used in a clinic, for example, to give you access to genomics while you're within the hospital. That's some years off, but that's the way the technology will be going. So as Heather said in her talk, we're trying to find out why some people respond to drugs and not respond. So you can see in the future that you might be able to test somebody's genome within a hospital setting before you give them either the medicine that they want to take or, for infectious disease, which kind of antibody they should take, antibiotics. And so what else have we been using these platforms for?
For Generation Scotland, we've been looking at methylation. So there's a thing called epigenetics. You can imagine DNA being a genetic code. We already look for variants and how those are linked to diseases, looking between healthy and patients with disease to identify how that links to phenotype. There are marks on the DNA that will dictate if genes are expressed or not. And so these methylation marks can be detected by either special protocols for running on the sequencers here, or by just feeding the DNA as it comes through the nanopore device. We've recently done some work using Generation Scotland samples to engineer how the data looks. But we're hoping to scale that up to a much bigger platform. So using the same technology as the small sequencer we showed you there, but on a much larger scale.
Thank you, Nikki. So that's a little bit about the kind of work we do within the lab. So we'll be very happy to answer any questions you might have. So thank you. Back to you, Sean.
I can see that we've had our first question in, and it's actually one that we get asked quite a lot in these events. Heather, where did you start your career, and where did your passion come from?
Oh, that's a very good question. So actually, I had a bit of a circuitous route, which is probably worthwhile mentioning. But I originally started off as a dentist and decided that I didn't want to do dentistry. I didn't want to spend the rest of my life looking down people's mouths. And I was much more interested in the brain. And the brain just fascinated me, which is why I started. It was actually, I switched from dentistry to biological sciences, and then I specialized in neuroscience as I realized that I was just fascinated how this ball of mush in your head can make you people and remember things and dream and all those other things. So yeah, it was a fascination with the brain that's driven me.
And what about you, Lee? Were you also from a nightclub? No?
No, so mine's very boring. That's why I skipped over it. So I was just really interested in genetics when I was at school. So I went straight to university to study genetics at Birmingham University. And then my first job out of university was at somewhere called the Sanger Centre, Sanger Institute now, which is where the whole the human genome was done. So that was my first job out of university. I didn't work on the human genome. I worked on pathogens, that causes tuberculosis and malaria and other diseases like that. And then from there, I came to Edinburgh to try, to like Emma, to try and get genomics more into a clinical setting and try to improve health. Again, within our team, generally, we have degrees, we've got PhDs. We're not academics. We're technicians. We're very technical. So we've got through that technical pathway because we like playing with equipment like this.
And I think, one question that we often get asked by students at kind of the stage that they're at, where they're maybe considering their options for after school, what would be your top tip for anyone that is interested in pursuing a career in medical research?
Is that for me?
Yeah, let's start with you.
Okay. Yeah, I would just say follow the things that you're really interested in. I mean, it seems an obvious thing to do, but I think you really just have to be passionate about what you're going to be studying. You're going to be studying it for a long time, and yeah, just to mainly follow your passions, really.
I think I'll probably want to just echo that as well. Certainly from my experience, it's like follow your interests, but also don't be afraid to try new things. Try things that maybe seem a little bit out of your comfort zone. Like my experience is, it's always better to try it than not try it and then regret not doing it, essentially. So basically just take as much opportunities that come your way.
I think for me, I think Heather's story and Kicky's story are quite interesting, that, you don't necessarily have to have this really clear career idea that you can be a bit circuitous. But when you're doing that, it's about trying to learn from each different job and role you've had. So things that we do within the lab, like stock control of reagents and using barcodes, those are all probably things that you would do within a shop environment as well. So you can kind of think about, how are your current skills and knowledge, how could you use that within a laboratory setting? And they're not always that different, really. And so to try and pull out that for any CVs and when you're thinking about work in the future.
To jump in there as well, like a lot of what we do in the lab, the protocols we follow are recipes. So if you like to bake, if you like to cook, then technician work is rewarded.
All excellent advice. Our next question is for Heather, and it's a really interesting one, actually. How do you think all of this new data technology will change psychiatry? Do you think we'll be able to predict mental illness in the way that we can other diseases?
Well, that's a really, really interesting question. And I would like to think that there's lots of optimism there. So there's, linking in with the kind of genetic side of things, unlike some of other areas of medicine, it's the genetics underlying the conditions are much more complex. There's not kind of a few points within the genome that indicate your risk. We are learning that. And certainly for things like major depressive disorder, which we study a lot, we know that it's a combination of lots of genes across the entire genome which confer your biological risk. And we're now getting to the stage that we kind of have big enough samples with genomics that we can start to formulate something called a polygenic risk score. So, and that's a risk score for each individual person about, how many of these small genes of small effect that all sum up to a threshold that have been linked to disorders in other populations. But I think it's also important to bear in mind that we know these disorders aren't completely genetic. We know that there are lifestyle factors, there are protective factors that are also important. And I think that's an important thing to get across, that you may well carry a risk, and that risk might well be able to be quantified moving forwards, but it's not the whole picture. And there are things that people can do to minimize their risk. And we just need to have a better picture of what those things are and what periods of people's lives are. It's important to kind of think about those interventions.
I'm sure people will be interested in hearing a bit more about some of the other studies that are running as part of Generations in Scotland as well. I know you touched on a few. Are there any others of interest that anyone might be keen to get involved in?
My particular interest is adolescent mental health. And so we have a new study which is starting, which is looking at loneliness in the digital world. So looking at the effects of smartphone use and looking to see whether online versus offline social interactions have the same impact or have the same kind of capacity for growth over adolescence. So that's a study that's about to start quite soon. And we're going to try and use smartphone-based apps to do a kind of very kind of quick fire recording of less than a minute, 4 times a day. What was your most recent social interaction? Was it online or offline? And a quick kind of assessment of how that made you feel. And that's a pilot study for a much kind of bigger study, which will be running across the whole of the UK on adolescent populations. And that has the advantage of having lots of rich data at lots of small time periods, which we can then tease apart to kind of form a pattern. So big data isn't just about longitudinal data over tens of years. It can also be data that's collected over a short period of time, but is very rich. So that's a study that we're just about to start recruiting for at the moment. We also have a study on sleep, which is looking at sleep over adolescence. And that's testing out some new technology whereby, in the past, sleep's usually been measured by the kind of actigraph watches. They're called actigraph watches, but they're essentially fitness watches that you wear, and you wear it when you sleep, and they give you information about different sleep stages, when you went to sleep and things like that. But a lot of people just don't like wearing a watch in bed, or they don't remember to put it back on, or the battery runs out, or the data's not uploaded properly. So we have some new technology which essentially looks like a smart speaker. It just sits in the bedroom and it just passively collects data using radar technology to look at people's sleeping patterns, which means that can be done over lots of different longer time periods. And you're not just restricted to a week or a week and a half. That could just sit there for months, gathering lots of rich data. And of course, we know that over adolescence, people's sleep patterns change quite significantly, but we just don't know what the boundaries of kind of normality for those sleep changes are. We don't know what's worrying. So there are a couple of studies that we have going on, which aren't actually related to genetics, they're just related to adolescent mental health. But I can talk about a genetic one as well, or we can move on.
No, let's hear about that. And then I've got a few more questions in from our audience as well.
Oh, great. So the genetic one of the genetic ones that I'm particularly involved in relates to the prescription data. So we're very interested in looking to see who is going to be a good responder or a poor responder to different antidepressant medications. And that sounds quite straightforward, but it's actually quite complex in terms of the amount of data we have to deal with, but also pulling out whether people are responding or not responding. So I gave the example of people switching a lot. That's just one example of somebody not responding very well. But we're also using some AI and machine learning tools to look more closer into the data that's recorded by GPs, for example, when they see a participant. And it's not just little codes that are in the data. We're actually using a technique called natural language processing, which is able to pull out, a bit like your ChatGTP, pull out pieces of text that we can then use to infer whether somebody is responding or not. And then we'll link that with genetics, with the idea that we discussed and that Lee mentioned also about the ideal being you could go into the clinic, you could have a blood test done, you could see which of your, which medication for antidepressant for depression might suit you best.
Cool. Sure.
Lee, our next question is for you, and it's how small is a single piece of DNA?
Oh, so if you unravel the DNA from one cell, it would be 2 meters long. So actually, DNA is a very long molecule, and it's an amazing feat that you can collapse a 2 meter long piece of string into something that's so small as a cell. And that's done by various proteins, chromatin it's called. But yeah, that's amazing.
Brilliant, thank you. I think one question that we've been sent in is around the trends that you have all seen in your careers so far and how the industry evolved. I think as part of Heather's introduction, we provided such an interesting overview of how things have changed since the kind of 1990s. Be great to hear from a few of you just about kind of what you've seen change in terms of trends.
I'm happy to go, but sorry.
You go ahead.
So I was just going to talk a bit about methylation. So that's kind of been something which has really changed over the past 10 years since I've been involved in this kind of research. So we previously thought that you have DNA and that doesn't change. But now we can look at the methylation patterns that Dickie mentioned, which is little chemical messengers which are along the DNA, which kind of indicates which proteins are going to be made. And we know that the environment can affect those chemical messengers. So things like smoking, for example, we now know that that affects those methylation patterns across the genome that you can then measure. And they are also, we know now, to be reversible as well. So it's a whole other area of research that has kind of really expanded. And it's a real indication of we didn't really know how the environment used to affect biology to some degree, but this is a real kind of intersection between genes and environment that we can now measure and use to predict disease. We can use to measure inflammation, for example. So yeah, that would be something I would say has really significantly changed, and I think will continue to change medicine.
Such a game changer.
Yeah, absolutely.
Emma, shall we hand over to you? I know you were about to.
It was pretty much kind of like what Heather was going to say there. I know certainly from my time at school, it was very much like, you've got your DNA, you've got your genes, and each gene does a certain thing. But as time goes on, I think they have sort of understood it is a bit more complex than just the fact that you've got genes. I don't know if you've ever heard the term like epigenetics as well. That's very much like Heather says, and it's a sort of a more of environmental influencer of how your sort of environment does have an effect on your genes as well. So yeah, I would say that's very much one.
I mean, now you can sequence. You can use a sequencer to answer a lot of different questions about how genes might be expressed and when they're expressed, and all the different things that affect that. So the picture is becoming more complete, but there's still a lot to be learned. And certainly when we started the sequencing service in the lab, 2011, I think it was, we had one sequencer. It was fairly low throughput, and since then we just every few years we have to buy new pieces of equipment to help answer the questions that different researchers have.
For me, it's all about miniaturization of the equipment. So you can see the MinION we showed, or handheld device, compared to this desktop version, and what the old versions used to be even bigger. So the amount of DNA you can use is becoming less and less. Also, the sensitivity is increasing. So we can now sequence single cells. So generally in the past, we'd get somebody's blood sample, and then we would sequence all the RNA and everything that's in there. That gives you kind of a global look, but now we can go to individual cells. You have a brain cell, a liver cell, and say, well, what's happening within this specific cell type? The other side of that then is that the amount of data we're producing is just increasing hugely. So the data handling aspects, I think, is going to be a real issue in the future, and it's just going to explode.
And what do you think will be the kind of next big leap forward? Do you have any predictions for the future?
So within the genomics, I think it's going to be linking genomics with imaging. So we're starting to see with that with some of the spatial work. So trying to see how specific cells in specific tissues communicate with each other and how they're different within a very close proximity.
Would anyone else like to make a stab at some predictions?
Kind of covered it.
Yeah.
I was thinking, the spatial aspect that Lee mentioned there. If you think about cancer within a tumor, there's been idea that all cancer cells be the same, and if you could identify what was wrong with those cancer cells, you could do something to treat it. But within a particular tumor, there's the cells themselves are different, and all this data that's going to be useful to help tease out what's changing and how that can be stopped.
Brilliant. Right, well, thank you all so much for joining us today. A massive thank you to Heather, to Lee, to Emma, and to Dickie for joining us. Our next session will be looking at your future in medical research, and we'll be joined by Dr. Alan Hughes from the University of St Andrews. Till then, you can check out more content on the Medical Research Scotland YouTube channel. Please do like and subscribe. But for now, I'd like to thank all of our fantastic speakers today, and for you for joining us too. And until next time, goodbye to you all. Thank you.